Welcome to my course on Microbial Biotechnology. We are in module number 1 and today we are going to discuss about classification and taxonomy of microbes. Before we begin, let us have a recap of the last lecture, lecture number 2, which was on the classification and taxonomy of microbes, and this particular lecture is also a continuation of that last lecture. There we had studied about various classification concepts starting from the 2 kingdom to 3 kingdom to Whitaker's 5 kingdom classification, and then finally we studied about the classification and phylogeny, where we studied about the basic taxonomic unit and the bacterial nomenclature including ICNP and binomial nomenclature. Then we had some discussion on the domain bacteria. Today we are going to continue the classification of bacteria based on modes of metabolism, then cell wall characteristics, and based on RNA sequencing. And then we will go on to discuss about the domain archaea and how it is classified. Then finally we will also study about the eukaryotic microorganisms like fungi, the protists, in today's lecture. Before we begin, let us discuss in brief about various modes of metabolism in organisms. We know that to support an organism's growth, including humans, we require energy and reducing power to drive the necessary biosynthetic reactions. Now, various organisms do these energy acquisition by different modes. So, some of these are the heterotrophs, which use organic compounds as their primary source of cell carbon. Then we have autotrophs, which use carbon dioxide as their major source of carbon, like cyanobacteria and algae. Then we have chemotrophs that generate energy or ATP using chemical bond energy. Then finally we have phototrophs, which produce ATP using light energy. Now, these kinds of modes of metabolism can actually be used for classifying organisms, especially bacteria. So, let us study about the classification of bacteria based on the mode of their metabolism. So, we can typically divide bacteria into 2 nutritional types. The first one is autotrophic and the second one is heterotrophic. Now, what are autotrophic bacteria? They are bacteria which synthesize their own food from simple carbon sources like carbon dioxide, and may be further classified into 2 types. Number 1, photosynthetic bacteria, and number 2, chemosynthetic bacteria. Photoautotrophic bacteria like cyanobacteria use light as their energy source and carry out oxygenic photosynthesis. So, here you can see an image, microscopic image of cylindrospermum, which is a filamentous cyanobacteria, and you can see that they can actually become very long in the image. These photosynthetic bacteria possess special type of pigment known as bacteriochlorophyll, which we have mentioned briefly in the last class. They also have other pigments like bacterioviridin and chlorobium chlorophyll. These pigments are located on spiral structures called chromatophores. And similar to higher green plants, photosynthetic bacteria synthesize carbohydrates by fixing atmospheric carbon dioxide. Some bacterial species carry out anoxygenic photosynthesis using hydrogen sulfide instead of water, and produce sulfur granules rather than oxygen gas, and this is the reason why we call them as anoxygenic. So, you can see here the reaction how hydrogen sulfide and carbon dioxide in the presence of light produce carbohydrates and release water and sulfite granules. So, in this picture you can see one species Chromatium oceni, which is a gram-negative purple sulfur bacterium performing anaxogenic photosynthesis. And due to this capability, these oceni can thrive in sulfur-rich, oxygen-poor environments in certain lakes and also in the sediments. So, here you can see the sulfur granules which are getting produced, and then these are the polysaccharide granules which are being produced in this entire process. Then these are the vesicles which contain oquinone, the carotenoid component, and it also has a flagella, as you can see in this diagram. Chemoautotropic bacteria are different. They do not use light for energy. Instead, they get energy from inorganic compounds like hydrogen sulfide, ammonia or iron, which they use to make their own food. So, we have examples of nitrosomonas bacteria which convert ammonium ions into nitrite ions, and then in the process it generates ATP in the presence of oxygen. There is another bacteria called nitrobacteria or nitrobacter. These convert the nitrate ions into nitrate ions, and in this process this also produces ATP. These reactions play a crucial role in the environment as they are part of the nitrogen cycle, as you can see in this diagram. Let us now discuss about heterotrophic bacteria. These bacteria cannot fix carbon dioxide and they obtain their energy and carbon from external sources. Two types of heterobacteria are there. The first one is the photoheterotrophs. They use light for energy and organic compounds for carbon. Some notable examples are green non-sulfur bacteria and purple non-sulfur bacteria. You have Chlorophyxus and Rhodobacter under these two types. Then another class of heterotrophic bacteria are the chemoheterotrophs. The chemoheterotrophs obtain both energy and carbon from organic compounds such as glucose. Those consuming dead organic matter are called as saprobes, for example Bacillus subtilis, and those feeding on living organic matter like human tissues are parasites, for example citrus pathogens. Some form beneficial partnerships with other organisms, and this kind of relationship is known as symbiosis. And some example of symbiotic bacteria we have here are the Rhizobia species. We can also classify bacteria based on cell wall characteristics. This 9th edition of Buzz's manual classifies the kingdom of prokaryote into 4 major divisions based on cell wall characteristics. So, you have here the divisions 1, 2, 3 and 4, and they also have some corresponding names like gracilicutes, which are gram negative and thin skinned. Then you have firmicutes, which are gram positive and have thick and strong cell walls. Then you have tenericutes and mendocicutes. Each of these various divisions have also certain classes. For example, division number 1 has 3 classes, 1, 2 and 3, and their corresponding class name you can see are Scotobacteria, which is gram negative non-photosynthetic bacteria. Then you have anoxyphotobacteria, which are also gram negative photosynthetic bacteria that do not produce oxygen. This includes the purple and green bacteria. Then you have oxyphotobacteria. These are gram negative photosynthetic bacteria that produce oxygen, and these includes the cyanobacteria. Under firmicutes, we have 2 divisions. The first one is Firmibacteria, which are gram positive typical Rothschild or cocci bacteria. Then you have the Thalobacteria, which is gram positive branching filamentous bacteria. This includes the Actinomycetes. And under the divisions Tenericutes, we have only one class, which is Molecules. These bacteria lack a cell wall and includes mycoplasma. Similarly, we have Mendocicutes, only one class, which is known as the Archaebacteria. These are bacteria with unique cell wall and membrane compounds. These are more commonly referred to as Archaea currently. One of the interesting classification of modern times is the classification of bacteria based on RNA sequences. Prior to these, DNA hybridization was considered as the gold standard for bacterial species delineation, which measures genetic relatedness by reassociating DNA strands from different organisms. DNA hybridization offers high precision in defining species with 70% or more DNA relatedness and a melting temperature of around 5 degrees centigrade or less, making it ideal for resolving taxonomic uncertainties. For this reason, it is called as gold standard for bacterial species delineation. However, despite its accuracy, DNA hybridization is a labor intensive, time consuming and costly procedure, and therefore, this has led to a decline in its routine use as more practical techniques like 16S RNA gene sequencing have become available and widespread. Therefore, new classification schemes based on RNA sequencing have revealed additional branches in the bacterial tree, and some of these align with Burgess system, example gram-positive bacteria, while others such as the spirochetes are genetically distinct and placed into separate categories. Why the 16S RNA gene has become the most widely used genetic marker for studying bacterial phylogeny and taxonomy? There are several factors responsible for these. Number one, 16S rRNA gene is present in nearly all bacteria, often in the form of multigene families or operons. This particular gene is having conserved function over time, making random sequence changes a more reliable indicator of evolutionary history, and it has adequate length, roughly around 1500 base pairs, which makes it well suited for bioinformatics analysis. Currently, there are approximately 100 recognized bacterial phyla identified through culturing or nucleotide sequencing methods. Let us see some of the prominent phyla and additional groups in the future in slides. So, let us start with Proteobacteria. It comes from the word proteo, meaning first, and it is the largest and most diverse bacterial phylum, including many well-known gram-negative genera like Escherichia coli. It is subdivided into alpha, beta, gamma and delta, and also epsilon. So, here you can see the example of Rickettsia under the microscope. Some of the notable pathogens under these are enteric pathogens like E. coli or its relatives. Then you have Shigella, Salmonella and Vibrio cholera, which causes gastroenteritis and cholera. And other significant genera are Neisseria gonorrhea, which causes gonorrhea, and Yersinia pestis, which causes bubonic plague. Then you have Rickettsias. These are intracellular parasites transmitted by arthropods like ticks, lice, fleas, mosquitoes, causing disease such as Rocky Mountain spotted fever. Then we have Actinobacteria. The phylum includes gram-positive bacteria with high guanine and cytosine content in their DNA. Many species exhibit branched filamentous growth similar to fungi, although not all do so. Their habitat, particularly they are found in soil. Actinobacteria are important decomposers, and some species are also aquatic. Their function includes mostly they are non-pathogenic, but there are certain pathogenic genera which includes Microbacterium, and some of the famous examples are Streptomyces, which produces over 2/3 of naturally derived antibiotics such as streptomycin and tetracycline, making it a very important species and genera. Then we have Mycobacterium. This includes pathogens like Mycobacterium tuberculosis and Mycobacterium leprae, which are known for their waxy cell wall and containing mycolic acids. And something very interesting, these actinobacteria contains problematic bacterium as well as bacteria which provides solutions to this problem. For example, it has disease causing pathogens, and it has also bacteria which produces antibiotics. The next in line is cyanobacteria. These are prokaryotic bacteria that can exist in unicellular filamentous or colonial forms. Historically, they were referred to as blue-green algae due to their pigmentation. They can also appear black, yellow, green or red depending on their pigments. For example, the presence of red pigments in some cyanobacteria contributes to the periodic reddish discoloration observed in the Red Sea, particularly during blooms of particular species. Cyanobacteria are notable among bacteria for their ability to perform photosynthesis using chlorophyll A, similar to algae and green plants, converting carbon dioxide into biomass. They played a crucial role in oxygen evolution around 3 billion years ago, significantly altering Earth's atmosphere by producing oxygen. So, in the introduction yesterday we were speaking about the role of microbes in climate change, and we can see that in the past also microbes has played crucial role in changing the climate of our planet. It is believed that chloroplast originated from an endosymbiotic event involving a cyanobacterial ancestor. Notable genera includes nostoc, oscillatoria and couple of others. Here we can see a tiny image of a marine cyanobacterium. Let us now discuss about Chlamydia. Bacteria in this phylum are small, obligate intracellular parasites that require host cells for growth and metabolism, similar to rickettsias. However, they are not closely related to rickettsias and are not transmitted by arthropods. Initially, Chlamydia was mistaken for viruses due to their small size and parasitic lifestyle. Chlamydia are now recognized as prokaryotic bacteria with a gram-negative cell structure. They reproduce through binary fission, confirming their classification as bacteria as against those of virus. There is a micrograph of Chlamydia pneumoniae over here in an epithelial cell in acute bronchitis, which is number 1 shows the infected, number 2 show the uninfected, and the chlamydial inclusion in number 3, and you can see the cell nuclei in number 4. So, chlamydia has a lot of medical significance. For example, Chlamydia trachomatis causes severe eye infections leading to blindness. Chlamydia psittaci causes ornithosis or parrot fever in birds, and this is transmissible to humans. So, it is kind of a zoonotic disease. Then we have Chlamydia pneumonia, which causes lung infections. Spirochetes. The phylum Spirochetes includes over 340 described species, most of which are gram-negative. Spirochetes are characterized by their helical shape and corkscrew movement, and you can see these helical structure over here in this electron micrograph of Treponema pallidum. They inhabit diverse environments from mud and sediments to insect digestive tracts, and can be pathogenic in the urogenital tracts of vertebrates. Human pathogens include Treponema pallidum, and you can see the picture electron micrograph of this particular pathogen here, and this causes syphilis and certain Borrelia species which are responsible for Lyme disease and relapsing fever. Borrelia species are transmitted by ticks or lice. Bacteroidetes are members of this phylum, are gram-negative bacteria with some species possessing flagella for mobility. In the human gastrointestinal tract, Bacteroidetes and Firmicutes are the two most dominant phyla, often constituting a significant majority of the microbiome. Bacteroidetes are crucial for breaking down proteins and carbohydrates, producing essential nutrients and energy. They also influence immune function and overall intestinal health. So, they are very important as a gut microbiota. One of the notable species are Porphyromonas gingivalis. This is associated with periodontal disease. Although 16S RNA is a very promising technology for classification, it has certain limits. Let us try to understand what are these limits of 16S rRNA gene sequencing classification methods. So, there is no doubt that it is a very valuable tool for bacterial classification, particularly in delineation of species, but it has limited power at the species level and poor discriminability for some genera. This is not very specific for every genera, only in particular genera they have very poor resolution. High sequence similarity does not always ensure accurate identification. This is something important to remember. For example, in Bacillus globisporus and Bacillus psychrophilus, we have over as high as 99.5% 16S RNA sequence similarity, but only 23 to 50% DNA relatedness. Similarly, in Edwardsella, we have species, we have around 99.35 to 99.8% 16S RNA similarity, but their DNA relatedness ranges from only 28 to 50%, even though they are distinguishable biochemically. So, several bacterial groups such as Enterobacteriaceae and rapid growing mycobacteria also encounter these resolution issues, emphasizing the need for DNA relatedness studies to overcome taxonomic challenges. So, while using tools like 16S RNA, we should be a little bit cautious and always look for the DNA relatedness in parallel experiments. So, this table lists some examples of bacterial genera and species with identification problems while using 16S RNA gene sequencing. So, you have various genus like Aeromonas, Bacillus, Bordetella, Burkholderia, then Campylobacter, and then some of these genus has many species which suffer from this kind of limitations, like in Bacillus you have the Anthracis, the Cereus, Globisporus and Psychrophilus. Let us now go to the next section, section number 2, and discuss about the classification of archaea. So, the domain archaea. Both bacteria and archaea are unicellular organisms, but they differ significantly in several aspects. So, if you look into this figure, you can see that there is a difference in these linkages. While archaea has ether linkage, bacteria has ester linkage. The phospholipid bilayer from bacteria and eukarya is different from that of archaea. You can see in this diagram. Now, let us try to list these differences as mentioned in this table. So, if we take the cell membrane, as I have already told you, you see that bacteria has ester linkages with unbranched fatty acids, while archaea has ether linkages with branched isoprene chains. In the cell wall, we have peptidoglycan in the case of bacteria, whereas archaea lacks peptidoglycan and it may contain pseudopeptidoglycan or other unique polymers. When it comes to the genome, bacterial genomes are small and less complex, whereas archaeal genomes are large and more complex. So, like prokaryotes, archaea share many similarities with bacteria, but are often more closely related to eukarya. So, they are in between the two, the bacteria on the one side and eukarya on the other side. So, what are the similarities with eukaryotes? The ribosomal RNA. It shares several RNA sequences absent in bacteria. Then when it comes to protein synthesis, it shows similarities in protein synthesis and ribosomal structures. Then comes the differences. Genetic sequences, unique sequences like this one, CA, CCZ in RNA is found. When it comes to the membrane lipids, we have distinct ether-linked lipids with branched isoprene chains, as we have already discussed in that figure. Then in the table we have mentioned that it lacks peptidoglycan and it may contain pseudopeptidoglycan or other unique polymers. Let us see the evolutionary perspective of archaea. Archaea are considered among the most primitive life forms, resembling early cells from 4 billion years ago. Modern archaea, these extremophiles, thrive in extreme environments similar to ancient conditions, such as hot anaerobic habitats which are rich in sulfuric gases and salts. So, how archaea are classified? Classification in the archaea domain is more challenging than in bacteria due to the more recent exploration and discovery of archaeal species. Recent molecular studies have identified additional phyla and more complex classifications reflecting the diversity and adaptability of archaea. Archaeal organisms are widespread across diverse environments, with some thriving in extreme conditions such as cold or high temperatures, while others adapt to more moderate habitats. Many archaea are extremophiles, thriving in extreme conditions such as high temperatures, high salt concentrations or extreme pH levels. We have the archaea divided into various phyla. The major phyla, Euryarchaeota, which includes the methanogens, the halophiles and several other diverse groups. Then we have Cranarchaeota, which includes thermophiles, acidophiles, and these are often found in hot and acidic environments. Let us discuss about the Euryarchaeota first. Euryarchaeota is a diverse phylum containing extremophiles with various physiologies and shapes, including rods and cocci. These organisms can be gram positive or gram negative depending on the presence of pseudomurine in their cell walls. The first is the methanogens. These are notable groups within Euryarchaeota producing methane from carbon dioxide and hydrogen, and thriving in oxygen free environments such as sediments, mud and animal gastrointestinal tracts. Methane production is crucial for their energy metabolism, and these species release over 2 million tons of methane annually, with about 1/3 coming from cattle rumen. For this reason, these species is also considered as very important species from the climate change point of view. Next come the halophiles. These are archaea which thrive in high salt environments, and hyperthermophiles which survive at extremely high temperatures. They require high salt concentrations up to 36% NaCl and oxygen for energy metabolism, thriving in highly saline environments like the Great Salt Lake, the Dead Sea and also in salt evaporation ponds. Some species known as halobacteria use a red pigment to produce ATP in the presence of light, contributing to the vivid coloration of red herrings, the Red Sea and the salt ponds. Certain halophiles also thrive in extremely alkaline lakes with a pH above 11. Hyperthermophiles. These are archaea adapted to grow at very high temperatures, as the name suggests. They thrive at temperatures between 80 degree and 105 degree and often tolerate high salt and acidity. They are typically found in volcanic waters, soils and submarine vents. For instance, thermoplasma flourishes in hot acidic environments such as near coal mines with a pH of 1 and temperatures around 60 degree centigrade. Researchers have also discovered hyperthermophilic archaea in deep sea sulfur vents where they survive at temperatures up to 250 degree centigrade and pressures of around 265 atmospheres. Crane archaeota. Crane archaeota or thermoproteota are primarily hyperthermophiles thriving at temperatures above 80 degree centigrade. They are gram negative and exhibit diverse morphologies including rods, cocci, filamentous and irregular shapes. These archaea thrive in extreme environments such as hot sulfur springs with temperatures around 75 degree centigrade and acidic pH levels ranging from 2 to 3 as well as in volcanic vents. Additionally, some of these are found in coal deep sea waters and polar seas, whereas where temperatures can drop to as low as minus 3 degrees centigrade. Let us now move on to section 3, which is a discussion on the eukaryotic microorganisms. Here we will be discussing about the fungi and the protists, their characteristics and their classification. Let us first define what are eukaryotic microorganisms. In this picture you can see a eukaryotic cell, and you can see here a nucleus which is enclosed by a clear nuclear envelope, unlike in prokaryotes. And then you have certain specialized structures like mitochondria here, and then you have centrosome, and then you have smooth endoplasmic reticulum and Golgi complex, and there you have lysosome and peroxisome. Now, eukaryotes may exist in microscopic form. So, these eukaryotic microbes are called as lower eukaryotes, which include protozoa, then microscopic algae, fungi and certain animal parasites. Typically, eukaryotic microbial cells have organelles as I have already discussed, such as cytoplasmic membrane, nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, vacuoles, cytoskeleton and glycocalyx. Some cells have also additional features like cell wall, locomotor appendages and chloroplasts. The fungi comes under the eukaryotic microbes. The kingdom includes a wide range of organisms like bread molds, yeasts, powdery mildews, cup fungi, smarts, rusts, puffballs and mushrooms. And these mushrooms are edible as well as some are poisonous, and we also use yeast for producing wine and bread, while there are other fungi which are actually involved in food spoilage. Some fungi are microscopic, while others can grow to over 2 feet in dimensions. Despite their differences, all fungi share several important properties. Number one, they are eukaryotic organisms characterized by complex cells with a nucleus, which we have discussed in the earlier slide. Their reproduction involves both sexual and asexual methods, resulting in spore production. Fungi exhibit growth in the form of hyphae. These are filamentous structures that make up the mycelium, or yeast, which are single cell forms with hyphae typically showing apical growth, meaning they grow at the tips. Fungi are heterotrophic organisms, meaning they do not perform photosynthesis. They can be saprophytes, symbionts or parasites, with some species parasitizing humans, animals or plants. Fungi absorb nutrients through their cell membranes via osmotrophy, although a few possess the rare ability of phagotrophy, which involves ingesting solid food particles. To break down high molecular weight substrates, fungi secretes various extracellular enzymes. Generally, fungi have sturdy polysaccharide rich cell walls, primarily composed of chitin in most species, while some may contain mannans, galactans or cellulose. What are the characteristics of fungi? So, you can see here one septate hyphae and one sinocytic or non-septate hyphae. So, here you can see certain divisions. These are the septate ones, and there are no any such divisions over here in these non-septate fungi. So, basically fungi are eukaryotic organisms with a thallus and which has different kind of complex forms. A mold thalli consists of long branched filaments. These are called as hyphae that forms a mycelium, and as already discussed in this diagram, some fungi have sinocytic hyphae without cross walls or septae, while others have septate hyphae with pores from cytoplasmic streaming. If we take up the case of yeast, they are unicellular fungi that reproduce by budding or spore formation and can form colonies, sometimes creating chains or pseudo hyphae. These are not real hyphae, they appear like hyphae. Yeast cells are larger than bacteria, typically spherical or egg-shaped, and contain eukaryotic organelles, but lack flagella and cilia. Fungal classification. Historically, fungal classification was based on structural differences and physiological or biochemical patterns. Today, DNA analysis and genome sequencing are used for understanding evolutionary relationship among various fungi. Fungi lacking a known sexual cycle are informally grouped as mitosporic fungi. These are often reclassified into a specific phylum once their sexual cycle is observed or when comparative genomics reveals a close relationship to other fungi. Fungi are generally categorized into 5 primary phyla based on their mode of sexual reproduction. For example, you have chitriod diomycota, which includes fungi with flagellated spores. Then you have glomeromycota that includes arbuscular mycorrhizal fungi forming symbiotic relationship with plant roots. Then you have zygomycota, which are known for producing zygospores during sexual reproduction, and we have ascomycota, known for asci sac-like structures that contain spores. Then you have basidiomycota, known for basidia, which are club-shaped structures where spores are produced. Let us discuss these in a little bit detail. Chytrids which are diverse group with around 1000 known species. They have a chitin in their cell walls, which is a characteristic polysaccharide of fungi. These chytridomycota are unique among fungi. They have flagellated reproductive cells that allow them to swim. They are mostly found in aquatic environments, both freshwater and in marine environments. Many are saprophytic, such as Rhizophylactis rosea, which decomposes cellulose in soils. Some chytrids also act as parasites on plants, insects and amphibians. Glomeromycota. These include arbuscular mycorrhizal fungi and are characterized by obligate symbiosis and asexual reproduction. Mycorrhiza denotes the close association between fungal mycelium and plant roots. Arbuscular mycorrhizal fungi create an extensive mycelial network in soil up to 20,000 km per cubic meter, absorbing nutrients like phosphate and providing them to plants in exchange for carbohydrates. Over 80% of vascular land plants form mycorrhizal associations, benefiting from improved nutrient uptake and enhanced plant diversity. Arbuscular mycorrhizal fungi also help control pests and pathogens. Some notable examples include Achelospora and Glomus. Zygomycota. It is a phylum of terrestrial fungi, includes around 1% of described fungal species. Common examples are bread molds, as you can see here in this picture, and molds on spoiled fruits and vegetables. So, basically this is a harmful species which is responsible for food spoilage. These heterotrophic fungi grow inside substrates using extracellular enzymes to dissolve and absorb nutrients. The cell walls contain chitosan and chitin, and they grow as sinusotic mycelium lacking cross walls or septae. Ascomycetes or sec fungi represent about 70% of all known fungi and display considerable diversity. Some notable examples include Saccharomyces cerevisiae, which we know call as the baker's yeast, then Morsella esculentum, then we have Penicillium chrysogenum and Neurospora crassa, which is a model organism like Saccharomyces cerevisiae. Ascomycetes have a vegetative structure that can be unicellular like yeasts or composed of septate filaments with multiple nuclei per segment. So, here we see the example of a septate hyphae with simple septal pores in Ascomycetes. The cell walls of Ascomycetes are mainly made of chitin and glucans with mannans present in many yeasts. Ascomycota and Basidiomycota are related, both having septate hyphae with cross walls and large pores for cytoplasmic flow. Their hyphae form mycelium to absorb nutrients from both dead and living organisms. The next is Basidiomycota, which includes about 30,000 species, and this represents around 37% of the known fungi. They can be unicellular or multicellular and are found in various environments. Notable members are mushrooms and puffballs, and you can see here a typical basidiocarp with cap, grill and stalk. The basidium, the reproductive structure, produces sexual spores called basidiospores. Vegetative structures can be unicellular or septate mycelium with cell walls made of glucans and chitin. Basidiomycota act as decomposers, form mycorrhizal relationships, and some are plant pathogens as well. Some examples include Cryptococcus neoformans, this causes lung infections, then Agaricus campestris, which is an edible mushroom, and then Amanita phalloides, which is poisonous death cap. So, you can see here Basidiomycetes from Ernst Haeckel's 1904 work, some beautiful diagrams over here of the particular species. Next comes the protists, which are mostly single celled, but perform complex functions akin to multicellular organisms. They thrive in aquatic environments including water, damp soil and mud, and may attach to surfaces or swim freely. The kingdom Protista, with over 64,000 species, is an artificial grouping due to its lack of common evolutionary traits. It includes unicellular eukaryotes and some multicellular forms like slime molds and brown algae. Protists are divided into two main groups, the algae and protozoa. Algae are photosynthetic protists. It can be unicellular, colonial or filamentous and vary widely in size from a few micrometers to 100 meters. They possess chloroplast and may contain additional pigments for yellow, red and brown colors. Most algae have a complex cell wall made of cellulose, contributing to the unique appearance of some types like diatoms and dinoflagellates. So, here you can see assorted diatoms 2 micron to 200 micron under the light microscopy. Euglenids such as Euglena have a flexible pellicle composed of protein layers and microtubules. Algae which often use flagella or gliding for movements, may have eye spots, stigma, you can see over here, to navigate towards light for photosynthesis. Microscopic algae are categorized into divisions based on chlorophyll types, pigments, cell coverings, stored food and genetic factors. So, they may be classified as Euglenophyta, Pyrophyta, Chrysophyta, Phaeophyta, Rhodophyta and Chlorophyta. So, Euglenophyta, these are mostly unicellular and exhibit motility by flagella found in fresh water. They do not have cell wall, contains pellicle, and they have pigments like chlorophyll, carotenoids and xanthophyll, and some are closely related to mastigophora, and some examples include Euglena. Then we have Pyrophyta, which are also unicellular and contains 2 flagella. They live in marine environments, and then they have cellulose or atapical wall, contains chlorophyll and carotenoids as pigments, and the cause of red tide or harmful algal blooms are ascribed to Pyrophyta. Then we have Chrysophyta. These are mainly unicellular, some filamentous in appearance, unusual form of motility. They are found in both freshwater and marine water, made up of silica, hydrated silicon dioxide called frustals, found in their cell wall. They have pigments like chlorophyll and fucoxanthin. Mostly they are the diatomaceous earth and mesocomponent of the planktons. Examples include Navicula. Then we have Phaeophyta, which are multicellular vascular systems, possess holdfast or root-like structure for anchoring. Primarily they are found in marine subtidal forests. Their cell wall contains cellulose and alginic acid and have chlorophyll, carotenoids and fucoxanthin, and they serve as source of emulsifiers and famous for providing alginate. Examples include Fucus sargassum. And then we have Rhodophyta. They are multicellular, found in marine intertidal forests. Cell walls include cellulose, have chlorophyll, carotenoids, xanthophyll and phycobilin. They are source of agar and carrageenan and other food additives. Examples include Gelidium. Chlorophyta have various forms ranging from unicellular, colonial, filamentous to multicellular. They are found in freshwater as well as in salt water, have cellulose in their cell wall, and apart from chlorophyll and carotenoids, they have xanthophyll as pigments, and these are presumed to be ancestral to higher plants. And examples are Chlamydomonas and Spirogyra and Volvox. Protozoa. Protozoa are a subgroup within the kingdom Protista, comprising approximately 65,000 species of single-celled animal-like microorganisms. Characteristics. They are known for their motility and varied form of movement, such as using cilia, flagella or pseudopodia. Reproduction. Pathogenic protozoa often reproduce asexually via simple cell division of the trophozoite, the active feeding stage. Many form cysts with a protective outer layer to survive outside a host, which can later release trophozoites when conditions are favorable. Protozoa can have complex life cycles, including both asexual and sexual phases, particularly in group like Epicomplexans. So, in these you can see various protozoans clockwise from top left you can see Blepharisma japonicum, which is a ciliate, then Giardia muris, a parasitic flagellate, then you have Centropixis aculeata, a testate or celled amoeba, then we have Peridinium willi, a dinoflagellate, then we have Caros carylensis, a naked amoebozoan. Classification of protozoa. There are various ways to classify protozoa. Let us discuss them one by one. The first is based on motility, reproduction and life cycle characteristics. We have Mastigophora. These are flagellated protozoa, some with amoeboid motility, reproduces sexually and have both cyst and trophozoite stages. Some examples include Giardia, Leishmania and Trichomonas. Then we have Sarcodina. These are amoeboid protozoa that reproduce asexually through fusion, mostly free living, and their examples include Arcella, Amoeba proteus and Foraminifera. Then we have Ciliophora, which is characterized by cilia, exists as trophozoites and cyst, mostly free living and harmless. Some examples include Paramecium, Blepharisma and Blantidium coli. Then comes the Epicomplexa. These are non-motile except for the male gametes, reproduce sexually and asexually with complex life cycles. All of them are parasitic. We have the famous examples like Plasmodium, then Toxoplasma gondii and Cryptosporidium under Epicomplexa. Another classification of these group is based on genetic analysis and genomics. Genetic analysis and genomics have led to a tentative taxonomy for clinically significant protozoa, dividing them into 3 supergroups not found in traditional Linnean classification. So, the first supergroup is excavata. These are single celled with flagella, modified mitochondria, often lack normal cellular respiration. For example, we have here trypanosoma, which causes sleeping sickness, and these are transmitted by CC flies. The next supergroup is called as Uniconta. These include amoebozoans, which are free living, self changing cells using pseudopods for movement and feeding. And we have here members like Entomoebia histolytica, which causes amoebic dysentery. The third supergroup is called as SAR, and it includes 3 subgroups. The SAR comes from the first letter of the 3 subgroups and is therefore a acronym. The first member of this subgroup are Straminophila. This includes photosynthetic diatoms, brown algae and water moles. Then you have Alveolata, which includes dinoflagellates, ciliates and epicomplexans. Then we have Rigeria, which contains foraminiferans and radiolarians. Epicomplexans are heterotrophic parasites with complex life cycles involving multiple hosts. For example, Plasmodium is an example which causes malaria. So, with this we come to end of this lecture, and then we will be discussing about the various methods and tools which are used in the classification in the next lecture. Thank you.